Hi guys, Mrs. H here. Today we're going to be looking at DNA replication. So by the end of this, you're going to be have a good understanding of how DNA replication occurs, the enzymes that are responsible for this process, and be able to give me some details about the process. You notice we have our vocabulary, which was part of our assignment yesterday, so that those words are going to be used in today's notes. So I hope you have a good understanding of those words. So DNA replication, exactly what is it and why is it so essential? Well, it's so essential because if I replicate my cell or I divide my cell, that new cell needs to have the instructions to perform life's tasks. So it needs the blueprint or the recipe book to function. And so DNA replication, or the copying of the DNA, is critical for the survival of the new cell that was made during cell division. And it's a very complex process because the genome is so big. And so let's go ahead and take a look at the process of DNA replication. So first off, what's the overall format of how this happens? So how does the DNA replicate itself? Well, it replicates itself in what's called a semi-conservative model. And what that means is that the original DNA strand, which has two strands, right, linked in, twisted together, they will separate themselves, and the two old strands are going to make a copy of themselves. So the newly formed strand has an original parent and a new strand next to it. So we call it semi-conservative because it conserves it has part of the original, and it's part new. So it's half old, half new. Some scientists did have other models that they believed. The dispersive, which basically meant that we make this in chunks or in pieces, kind of like having a model house where the parts are all there, but they have to come and put the parts of the house together. We know this isn't true based on some research and some studies that they've done. We found that the dispersive is not how it's made. The other was the conservative, is that when the cell divides, we have a whole new strand of DNA, that there's no original present. And we also know that that is not true. This is not how this occurs based on the science experiments that scientists performed. And if we look at this, the three models side by side, with the gray being the original strand and the red being a new strand, you have conservative, one old, one new, dispersive, part old, part new, but in chunks, and the semi-conservative, one whole strand old, one whole strand new. And really, how do we know this occurs? We don't necessarily see it as evident in our first replication, but when we get into that second replication, we can see where this semi-conservative replication occurs. So with DNA replication, enzymes are critical for the reaction to occur. So this is a chemical reaction. When we have DNA replicating itself, we are having reactions occur. And I know we think of a chemical reaction as this big explosion, but it's not necessarily a big explosion. You have chemical reactions occurring all over the place in your body. Whenever your DNA is replicating itself, it is a chemical reaction, and so it needs enzymes to help facilitate this. And so if we look, there are the DNA topoisomerase, DNA helicase, the single-strand bonding enzyme, or the single-strand binding, it's a prefix or a tense issue, so it doesn't matter. It's all the same thing. We have DNA primase, DNA polymerase, ligase, and then again, DNA polymerase. So we're going to go ahead and go through the roles of these enzymes. in this process. So the most important one to DNA replication is DNA polymerase. Polymerase is responsible for adding nucleotides to the complementary strand, to the template strand. And so without this, you're not going to have DNA replication occur, because we need to be adding nucleotides, right? If I want to make a DNA strand, I need to have nucleotides. So I need to add them in some way. And so DNA polymerase is the one responsible for that. And so what does it do? So it takes the template. So we have the two strands, and we're going to take the template strand, and the DNA polymerase is going to add nucleotides to the template strand following the base pair rule. So what are some features that the DNA polymerase has? DNA polymerase can't initiate the DNA replication. So we need something to help it get started. And so we need a primer, which is a short strand of nucleotides, which the polymerase can actually attach to. DNA polymerase needs a template strand, so it needs a strand to read the sequence so that it knows how to add nucleotides. So it's basically, it needs a directions strand. The DNA polymerase can only move in one direction. It can only move from the 3 to 5 direction on our template strand. And we also know that DNA has to make sure it's doing it correctly. So we need to make sure that it's proofreading for mistakes. So DNA polymerase requires energy. So in order for this to happen, we do need to have some kind of energy, and that energy actually comes from the breaking of these phosphates off of the nucleotide. And actually this is called, when we get into energy and ATP, we will see that when we break phosphates, we're actually going to release energy. And so that's what happens. We have some extra phosphates onto the nucleotide. And so to get energy, we're going to break the phosphates off so that it can A, get energy, and then B, add the nucleotides to each other. Another interesting thing that you should know is that DNA replication occurs along multiple locations along the DNA strand. So it's not like we start at the beginning of the train track and just run all the way down. We break it up. And so there are multiple locations that DNA replication is occurring. And so it allows this process to happen more quickly and more efficiently. Now, in a prokaryotic strand, it actually only starts at one end, but it goes in two directions because prokaryotic cells are circular. And so they start at one spot, and then it actually wraps around in the circle until it meets the other end. Where eukaryotic, because it's linear, will actually have multiple places along the strand that it will be replicating at. These locations are called the origin of replication, and when that happens, it actually is going to create a replication fork, because it literally is, there's a fork in the road, that's where it comes from. And so when this happens, when they're separating and that fork, we actually get a replication bubble. So in this little sphere, or in this bubble, that's where the DNA replication is occurring. And so you have multiple bubbles, multiple replication forks occurring along the DNA strand on a eukaryotic cell. Prokaryotic cell, you're only going to have two, because they're going to move in opposite directions down around, and so you only have two bubbles. In order for the replication fork to occur, so to have that organ so that the DNA polymerase can attach itself, I need to have helicase and I need to have the single-stranded bonding protein. So the helicase, it will bond to a site of origin. So not just some random spot. It's a very specific spot on the DNA that it binds to, and it basically untwists, and you can actually see it right here. So we can see it untwisting the DNA. And so we want it untwisted so that we can actually have access to those hydrogen bonds. And so then what's the next thing we need to do? Or sorry, this right here, wrong thing. This is not this one right here. This is the helicase that's breaking it. This typoisomerase that we'll talk about in a second. Sorry, got ahead of myself. So then in order to keep this open, this is the single-stranded bonding agent. So we want to break those hydrogen bonds. That's what the helicase is doing. We want to keep it untwisted so we can break those. That's the typoisomerase. But now after we did all that work, we don't want to undo our work. So how do we keep it propped open? And that's the single-stranded bonding proteins. Sometimes it's called enzyme. Remember, you can call an enzyme a protein, but not all proteins are enzymes. But you can, in this situation, flip between those two words. So now what happens? So now we have those primase, that's the primer. So these primases are going to attach to the DNA strand so that the DNA polymerase can come and attach itself to the DNA molecule. Most primers are 5 to 10 nucleotides in length. And again, it's going to be on the 3 end. We're going to still be moving in that 3 to 5. So we're at the 3 end of the DNA strand. So if you notice, these are not opposite each other. Okay, they're kind of staggered because this has to be farther down, okay, along the strand to get at the 3 end side. This is the 3 end, so it can, it's going to be closer to the replication fork. So that's where that leading and lagging strand comes from. So the leading strand is going to be the strand that we can continuously move the DNA primase down the strand, continuously adding the nucleotides on the 3 end. So if this is the replication fork right here, if this side is the 3, right, and this side is the 5, this side can add the nucleotides, the primer can be attached, the DNA polymerase can be attached, and it's just going to zip on down the line. This cannot do that. It's like putting the key in upside down. It won't fit. So I have to flip it over. Well, how do you flip over a DNA strand? You can't. So you got to work in segments. You got to start farther down along the line. You got to start down at the 3 end and work your way back. And then back, and then back, and then back. And so it results in these scenarios where you don't have a continuous strand being made. That's where that Okazaki fragment comes from, because they're being made in those fragments. It's the primase is farther away from that replication fork, and so that's how you know that it's the lagging strand. You look, is the replication fork and the DNA polymerase, are they far away from each other? And if they are, then you know it's a lagging strand. Then you know that strand is going to have Okazaki fragments. But here's the thing, when I create those Okazaki fragments, I'll actually get little gaps, okay, that have to be filled in, okay? And so in order to fill in those gaps, we also need another protein to come in and fill in those gaps. So on the, I forgot where it's going. So when we're running through our leading strand, we actually have a DNA polymerase 3. The DNA polymerase 3 is one of the molecules, is the sliding clamp, which is ring-shaped, and it keeps it attached to the DNA strand. Okay? It's also there to make sure that mistakes aren't being made. So it makes sure that DNA replication is occurring in the order and in the fashion that it should, so we don't make mistakes. Because if we make mistakes, then our new cell is going to have that mistake, and so then we have genetic mutations. I had referred to this earlier. This is the topoisomerase, and that's the structure that helps the DNA strand keep unwind so that we can break the hydrogen bonds and so we can add the nucleotides. So if we look at this model, we actually can see all of those enzymes present. We have the topoisomerase, we have the single strand bonding enzyme holding them open. Here you have the primer, the primase attached. Then you have the helicase that's actually breaking those hydrogen bonds. You have your DNA primase that's running down the leading strand and the lagging strand, adding those nucleotides together. So when we're looking at cleanup, the DNA polymerase is also responsible for cleanup, and what they do is during this process, RNA nucleotides are added temporarily. And so that's what happens on the lagging strand and that primer. That primer actually adds RNA nucleotides. So now that the DNA molecule is really set to go, we need to go ahead and get rid of those, and those just kick them off and get rid of them. And so then we're able to fill in the gaps and put in the DNA nucleotide where we need to. How has this happened? This is the DNA ligase. So DNA ligase comes in and fills in those gaps and gets rid of those RNA molecules that were put in there just temporarily when those Okazaki fragments and those primers were added. So if we look at this as a whole, this can be actually described in three processes: initiation, elongation, and termination. Initiation is when we're, separating the strands and untwisting and unwinding. The elongation is when we're adding our nucleotides, and then the termination is when we're filling in the holes and we're proofreading for errors. All of these are happening simultaneously. Maybe you can say, well, initiation has to happen first, and that is true. But remember, you're having DNA replication occurring all over the place on the DNA strand. So once it's been initiated, you have elongation and termination happening at the same time. And so it's not necessarily a linear sequence. In your traditional fashion, which you think like initiation, elongation, termination, initiation, elongation, termination. It's maybe a cycle where you have overlapping cycles. And so let's go ahead and review putting all of these enzymes together. So what do we have first? We have the topoisomerase untwisting the DNA. And then we have the helicase. They're going to break the hydrogen bonds between the complementary bases. So those base pair rules, those bases are going to break open. Then you have the single-stranded bonding. They're holding open and preventing that all that work from being undone. And then you have the primase adding a primer, remember, which is the RNA that allows for the DNA polymerase to attach itself. So once those three things are in place, we can go into elongation. This is when the DNA polymerase is sliding down the DNA strand, either leading along in a continuous manner or lagging behind because it's making those fragments. And the DNA polymerase is responsible for adding those bases. How do we know what bases it's adding? Well, if I have an adenine, what can bond with adenine? Thymine. So that DNA polymerase is going to add a thiamine there. And so they follow the base pair rule. Also to remember that we always add the nucleotide on the 3 ends. So remember the direction. Remember I talked about the direction. Where is the 5 and the 3? Because that's going to tell you what direction that DNA polymerase can run. Is going to run towards the replication fork, or is it going to be running away from the replication fork? So that is your elongation. And then you have your proofing, your termination. So your lagging strand has to go clean up those holes. So that's where we have that ligase kicking off those RNA nucleotides and putting in and replacing them with DNA nucleotides. And then we also have the DNA polymerase double checking. Did I add the right nucleotide? Did I add the right thing? It's like your proofreader, it's your editor. And then what do we end up at the end of this? We have two completely disassembled DNA strands that are semi-conservative. We have that template or the old strand with the new form nucleotides on the other strand. So again, it's semi-conservative. Here are some helpful videos. The Ed Talk actually is an animation. It's a fantastic animation that describes and shows you what's going on, what it would look like in not real time, but in an animated form. Mr. Anderson and Mrs. Riggs, they actually have really good videos that describes DNA replication as well. It's not rocket science. Now, it's not rocket science doesn't go into as many of the enzymes that I have, or Mr. Riggs, or Mr. Anderson and Mrs. Riggs, and then obviously my video that you just were finished watching here. So I hope this was helpful. I hope you have a better understanding of DNA replication. Remember, what's the takeaway? I want you to understand that DNA is semi-conservative, that there are multiple enzymes and what the role of each enzyme is in DNA replication, and that this is occurring over several locations along the DNA strand, and that this is a very complicated process because of the complexity of DNA. So I hope this was helpful. If you have any questions, please don't hesitate to reach out to me. Bye, guys.